Good afternoon. Thank you for the introduction. Thank you for the organizers for asking me to participate. Thank you, Rick Adams, for kindly recommending me. Today, I'd like to talk about a topic near and dear to me, the treatment of advanced malignancies, human cancers, the scourge that kills more people than any other disease in the population less than 78 years of old age. So we are now really looking at the leading cause of death in America. Cancer, as we know it, may be very different from the way we've approached it. And so what I'd like to talk about today in the context of cancer as a window on human biology and the studies that we conduct as a way to really explore new discoveries that will allow us to understand aging and many degenerative diseases. So cancer, in a way, is a window. Now, my premise is that the future of cancer research lies behind us, that we may have many discoveries that we came upon many years ago that nonetheless will lead us to better outcomes. Now, Clifton Leaf, several years ago, published a paper, the lead article in Fortune magazine, and the article described his disappointment that we were losing the war on cancer and some suggestions on how to win it. Now, you can imagine that this article met with quite a lot of consternation on the part of the academics who were quite sure that everything was going along swimmingly, and they weren't very happy to hear all this. Well, it turns out that I think Clifton raised a lot of very important questions, so I think we ought to address them today. Why haven't we cured cancer? I missed, unfortunately, Dr. Earle's interesting talk, but we can explore the oceans, we can go into space, and yet we have very little advance in this important disease. So let's go and look at the questions that we pose. Have we not dedicated adequate resources? Have we allocated resources inappropriately, ineffectively? Is cancer too complex for us to figure out? Is it beyond the ken of the human mind to understand this disease and better control it? Are the working models, those techniques that we use to study this disease, not appropriate for the disease at hand? And I guess finally, is cancer curable? Well, I'd like to answer these questions that have vexed cancer researchers for many, many generations. The answers are no yes, and maybe. I'd like to thank the organizers for allowing me to come today. Well, let's look at these things one at a time. Have we failed to dedicate the necessary resources? No. I would say that today, inasmuch as we spend over $200 billion a year in the United States on the treatment of cancer, between the direct cost of cancer therapies and the lost productivity and other expenses, $200 billion is a pretty good amount of money. I guess today, in today's political discussion, it doesn't sound like much, but it seems like a lot to me. Over 1.5 million peer-reviewed articles are today in the literature in cancer alone. That sounds like a large number of studies. And the National Cancer Institute has the single largest budget of any institute in the entire National Institutes of Health. Well, have we allocated resources ill-advisedly? I'd say yes. The research support today reflects the fact that PhDs answer interesting questions, while MDs solve medical problems. Yet the majority of the National Cancer Institute's funding today is dedicated to PhD scientific investigation. As a result of this, these scientific discoveries are not translating into the improved clinical outcomes that we are all hoping for. The taxpayer dollars that are going toward better outcomes are not being seen. Only 1 out of 7 clinical trials today. You all hear about the trials at the City of Hope, and you see all these great things we ought to be signing up. But in point of fact, only 1 out of 7 trials reveals that drug A is better than drug B statistically significantly, and more disturbingly, only 1 out of 14 clinical trials provides a response rate that gives survival of 50% or greater to the winning arm. Usually days, sometimes weeks, rarely months, and never years. Survival for advanced cancers today are unchanged in 50 years. In point of fact, if anyone in this audience was diagnosed with advanced cancer today, the chance of your surviving 5 years is no better than it would have been when my 1955 Buick Special, model 41D, rolled off the assembly line in Detroit, a city that used to make cars. If Donald Trump were to confront these issues, I'm sure you know what he'd say. The cancer research community ought to really take more seriously who they're working for. Is cancer too complex? Is it beyond our ability to study it? Is it just that this is so complex and so complicated? And I would say no, not necessarily. It's just that we like to make it seem that way. Every one of these bubbles inside this molecular cascade is a PhD career in the making. Every one of these little T-bids and granzyme Bs is a discovery to be made by someone who spends a whole career figuring it out. So they're looking at the single nucleotide polymorphisms in caspase-9 where it's binding site to the inhibitors of apoptosis protein as they downregulate through SMAC Diablo and influence caspase downstream events. And at the end of the day, I don't care. Because at the bottom is rest in peace, cancer. That's what matters. We are, as MDs, empowered to kill cancer cells. I don't care how I kill them. What I care about is that they die. And killing cancer is what I do. In my laboratory and in our group, all we're interested in is clinical results. Some smart guy can figure it out. Are our laboratory models inaccurate? Is it possible that we have in fact gone down the wrong pathway? Yes, absolutely. There's nothing more I can say today than how strongly I believe that we've done this wrong. Contrary to the long-held belief that cancer represents excessive cell growth, it is now abundantly clear that actually cancer represents a deficiency of cell death. Cancer doesn't grow too much. It dies too little. And in fact, the way that cancer is winning this battle isn't by having lots and lots of children, but by staying in the gene pool long enough to have an extra generation or two, and the generation has another generation, and after a while, we accumulate tumors. We don't grow them, we accumulate them. So cancer is not driven by its proliferative capacity. It isn't DNA and RNA and all that informatics. It's longevity, survival, and energetics. Life is energy, and cancer is driven by better batteries. Now, loss of cell death in a cancer is a little bit like an aging population that refuses to grow old and die so as to make room for the next generation. I think you get the idea. There is an ebb and flow, a yin and yang of life that we all should adhere to, and cancer breaks the code. The social code that the cancer cell lives under, where the body as a whole, the metazoan organism, gives that cell the opportunity to stay alive as long as it does the work of the body as a whole. This is a social contract like Locke, the idea that the cell is part of the whole. And when a cell is mutated or damaged or can no longer perform its duty, it must die. And the cancer cell says, I'm going to stay alive. I'm going to continue this process whether I'm working or not, whether I'm doing my job or not. I'm going to stay alive as a cancer cell, and as a result, you get all of these aberrant, mutated, abnormal cells. And if you put it in the human idea, if you had children and more children and great-grandchildren, and all of them were living in the house, well, pretty soon one of them jumps out the window and goes somewhere else, and that's metastatic disease. But it isn't happening by growth, it's happening by accumulation. Cancer cells accumulate to the numbers that we see as a tumor. Now, is cancer curable? Is this a disease that we can actually cure? Well, maybe. But it isn't going to be trivial. And the trouble with scientists today is that they're reductionists. They want to take this incredibly complicated problem and turn it into some sort of little puzzle. It's just a nice little collection of DNA base pairs running along the genome, and we're all going to figure it out. And therefore, we now have anointed the molecular biologists as the only scientists in cancer research. They're the only guys in the game. You can't get funded if you're not doing genetic analysis. You can't get funded if it's not Affymetrix platforms. It's all got to be DNA and RNA. So now we have companies offering DNA and RNA analysis to tell cancer patients what treatments to take. Venture capital-backed companies all over the country are selling this. We're going to run your DNA. We're going to tell you what makes up all the genes that make you, your cancer, run this way and that. We're going to treat you accordingly. Now, I don't buy into that. But if you're seriously interested in getting that, if anyone needs that sort of service, there's just one we recommend. eHarmony.com Oncology. When you're ready to find the treatment to save your life, we'll ask your tumor 29 genomic questions, and we will know exactly what treatment you should take. Or not. I don't buy in. I think that cancer is demonstrably more complicated than all this, and it's not just a collection of DNA base pairs. It is a living entity. It's the interaction of all of the various features, the lipids, the proteins, the epigenetic phenomena. It isn't simple. And all of this really lovely stuff that everyone was selling came to a crashing halt last Friday in the New York Times, when a study written by Gina Collada examined something that we in the medical oncology literature, at least I, have been following for a long time. It turns out that everyone was absolutely drunk on DNA, and Duke University was having a kegger. So Duke University decided they were going to go out and sell a gene profile to tell people how to treat their cancer, and it was a bunch of hooey. It's a bust. And it busted in disaster and lawsuits because it was made up. The lead investigator wasn't a Rhodes Scholar. He's been discredited and fired. His senior investigator has been demoted because they made it up. It all sounds so awfully good that it was too good to be true. It really was. With the dawn of the genomic revolution, we forgot at our peril that cancer is a bioenergetic metabolic process. We are living, breathing entities. This is cancer. And if you want to get really frightened, I'll magnify that. Because that stuff is really, really hard. Human biology isn't easy. It's complicated. It's crosstalk. It's convoluted. The interacting pathways, the redundancies, the complexities, the promiscuities of all of these events from one hydrogen up in the corner where a particular pentose shunt event leads to something downstream, ultimately changing the way a nucleotide is synthesized. All of this interacting phenomena is very, very hard, and we should have known it. Edward O. Wilson said in 1998 in Consilience, "The cost of scientific advance is the humbling recognition that reality was not constructed to be easily grasped by the human mind. It wasn't put here as a puzzle. You can't call up Will Short in the New York Times and ask him for the word you're not getting. It's that hard. It really is that hard. And we must be humble. We must stop trying to force human biology to behave according to our molecular dictates. And we must be humble enough to be instructed by the extraordinary forces of human life. It is extraordinary. You can't even begin to imagine how interesting, but it is not trivial. Here is where life takes place. If you ask many people what life is, they'll tell you it's precise self-replication, the ability to make another one just like you. Not so. Life is energy. Life is the directional or purposeful use of energy, an energetic process that goes from one point to another, a vector. Life starts not in the nucleus. The nucleus is the tombstone of the cell, and your epitaph is written in DNA. Life is energy, and the energy of life is what drives all of us as entities and cells. And if you can capture that energy and turn off the energy of cancer cells, then you've got a cure. We have not spent any time looking at this. We've been so dedicated to this gene revolution that you read about in Time magazine. I'm sure everyone in the room thinks it's the breakthrough that'll make all the difference. Well, it isn't making any difference, because life is very different from its informatics. Life is energy, and the mitochondria of the cell, that little pink disc at the top, is where it all happens. That's where glucose and acetate is turned into energy, and energy is put to a purpose. So if you want to cure cancer, if you want to treat this disease, you take that energy and you turn it off. And you do so by damaging the cell in a way that makes it want to die. And when it dies by one of many cascades. All roads lead to cell death if you do it right. One's called apoptosis. That's a very orderly process whereby the cell releases cytochrome C activates the apoptosome, and the cell kind of goes away. There's a more abrupt form called necrosis, and that's in the middle. And then there's a sort of middle point where the cell sidles up and back and tries to decide. It's a kind of Shakespearean Hamlet-like experience, whether to die or not to die is the question, Is it nobler? Anyway, the point is that here is a process that the cell goes through, and if it dies, if we win, if the equation goes our way, we cure the patient. In our laboratory, you can isolate cancer cells from patients. You can pull out these little clusters, these little extraordinary vessels. They look like, up on the screen, they look like Australia and New Zealand. Well, what that really is are about 100 or 70 cells all clustered together with blood vessels and stromal elements and inflammatory cells and cytokines and this whole ecology of cells talking to each other. And if you can introduce an injury that causes these cells to die, then those cells will involute and that patient will respond. So if these are your cancer cells on drugs. Cancer cells that are suffering the injury I talked about in real time, in 72 to 96 hours, will undergo the process of apoptosis, necrosis, and autophagy that leads to what we know as a clinical response to therapy, a bioenergetic metabolic cessation of life. So will we continue, as taxpayers, to dedicate our resources to a single solitary pursuit, the study of cancer informatics, the Human Genome Project, $3 billion and counting? Will we continue to do one form of science, or will we return to the work that led us to all the discoveries prior to 1953 when we figured out the structure of DNA? The cancer puzzle is very complicated. Now, for any of you who have ever sat next to a child at one of these restaurants that gives out crayons and a maze, you know that the kids usually figure these out quickly. They cheat. So do I. I go to the center of the puzzle and I look for the answer. I look to see if the cell died and I work backwards. It's reverse engineering. And what I do is figure out the answer and then I go back to what molecular enzymatic process led to that cell to die. And if I can find that, I've got the answer to my patient's problem. Does it work? Well, here's an example of one patient who came to me 7 years ago with widely metastatic non-small cell lung cancer. We took a biopsy of her tissue, placed it in the test tube environment, and studied it as I mentioned. She had a complete response, and 2.5 years later, it was so remarkable we got this donation from the Angels Foundation. I just talked to her last week. She's up on her walnut farm picking walnuts in Sacramento, 7 years later. Actually, I have another patient here, right here. I have another patient in attendance, similar story. That Merwin right there is 3 years out. And so the point is it does work, and we can do it. So will we continue the paradigm of genomics that has led us into this morass, this informatics only, as I said, it's picking treatments appropriately, but will we move on to the next, or stay with this paradigm that I feel is failing us? Genomics or metabolomics? The field of human cancer as a metabolic, bioenergetic phenomena, the real answers, in my opinion, are going to be found by those investigators who years ago studied these phenomena and discovered how we could chemically and biochemically, the Hans Krebs and the Otto Warburgs of the world who made these extraordinary discoveries, those are the pioneers. And I'm quite convinced that the future of cancer research lies behind us. Thank you.